Well, the system, every time I try plugging the screen, is crashing my computer, so I'm not quite sure what's going on. So, it looks like I'm going to have to do it without projection, unfortunately. Let me get this restarted so I can bring up things as necessary. So how's studying going? Nervous smiles, nervous laughter? Yeah. Making sense? A lot of material, obviously. How's your note cards coming along? Scared to touch it. Well, see? The note cards are interesting that way because you do realize how much you know when you start organizing the note card. So I recommend making a separate note card that you use to map out what will be on your actual note card. I think that's a good way to go, a good approach to go. But I give you the note cards so that you learn something about organizing thoughts, because I think you'll discover you're not going to use the note card very much at all. Most people don't. Yeah, because you already know it. Once you write it down, you know it. Which is why writing it down is important. That's why I try to get people to write it down, you know. Okay. So the exam, as you well know, is on Wednesday morning at 9:30 in our usual classroom. And I'm going to try one more time here and see if I get anything. And let's see. System preferences. Displays. Color. As soon as I plug it in, it just crashes, bigger than Dallas. All right. Well, I give up. All right, so where should we start? Day one. Oh, I'm sorry, yeah. Fatty acid synthesis. Okay. So, unfortunately, I can't show you fatty acid synthesis, which would be really nice to be able to do. But let me describe it to you. In fatty acid synthesis, you have the same 4 steps in fatty acid oxidation, except for they're backwards. And you, in addition, have another step involving the enzyme acetyl-CoA carboxylase. So acetyl-CoA carboxylase is the enzyme that you may recall that takes carbon dioxide and puts it onto acetyl-CoA and makes malonyl-CoA. So that's a molecule that has 3 carbons. Okay? That malonyl-CoA is converted into malonyl ACP, and another acetyl-CoA is converted into acetyl ACP. And those 2 molecules are the starting material for fatty acid synthesis. Fatty acid synthesis, of course, occurs in the cytoplasm. And in the synthesis process, the 2 of the 3 carbons of malonyl-CoA are added to the growing fatty acid chain, starting in this case with acetyl group. And the carbon dioxide that was put on is lost. So at the end of that addition, and so, well, let me back up. So acetyl-CoA carboxylase is the only enzyme in fatty acid synthesis that's regulated. It's regulated by the end product of the synthesis, which is palmitic acid or palmitate or palmitoyl-CoA, and it is that inhibits it. It's also activated by citrate, and citrate, you remember, was used in the shuttle to move the acetyl group out into the cytoplasm from the mitochondrion. Okay. All of the enzymes of fatty acid synthesis after acetyl-CoA occur in a complex that we call fatty acid synthase, and that's located in the cytoplasm. And That complex exists across a wide variety of cells, all the way from, I think, bacteria to humans. Okay? So it's a very, very long evolutionary history. But it's in those reactions that the fatty acid is made. The first reaction after the condensation has happened is a reduction of the keto group on carbon number 3. So the keto group on carbon number 3 is reduced to an alcohol, and that reduction requires electrons from NADPH, again catalyzed by fatty acid synthase. In the second step of the process, the water is removed from that using the OH that was created in the previous step and a hydrogen on carbon 2. That removal of water results in the loss of 2 electrons and creates a trans double bond, okay, between carbons 2 and 3, just like we saw in fatty acid synthesis. And in the last step of the process, the double bond of the that was created by that last step that I just mentioned is reduced by the protons and electrons from NADPH again. And that gives you a fatty acid then that has 2 more carbons than what you started with. And that fatty acid with 2 more carbons becomes the substrate for the next round of replication. Next round of synthesis. And that synthesis occurs all the way up until 16 carbons are achieved. That gives you palmitoyl-CoA, or palmitoyl-ACP, which is converted into palmitoyl-CoA. And at that point, the fatty acid synthase stops. Now, what I didn't tell you in class, I don't think, but I'll tell you here, and you're not responsible for, is that to synthesize fatty acids longer than 16 carbons, this occurs in the endoplasmic reticulum using enzymes called elongases. And elongases are enzymes that add 2 carbons, very much like what we saw here. There's also enzymes. There are also enzymes in the endoplasmic reticulum that desaturate, that is, take and make double bonds in the fatty acids, and those are called desaturases. And I don't believe I mentioned that in class either. Yes, sir. Malonyl ACP and acetyl ACP. So the acetyl group is the starting material, and onto that, 2 carbons of the 3 of the malonyl group are added. Okay?
And when that fatty acid, when the 2 more carbons are added to the fatty acid, it's just ready for the next round. The next round must be to get that started, or is it just
Nope, because now you have a 4-carbon starting material onto which 2 more carbons are added, just like they were on the first one, and it just keeps going on until you get to 16. Yep. Is that a question, Nick? No. Andrew? So our DNA histone complex is considered supercoiling. Supercoiling, that's not easy to say, the answer to that question, Andrew. Supercoiling exists anytime you change the number of base pairs per turn from 10.5 to something else. And so if making of that chromatin for using the histones in the DNA results in a change in that 10.5 base pairs per turn, then you would say, yes, it is supercoiled. And if it does not, then it's not. No. No, it happens in eukaryotes as well. Definitely. Yep. I'm sorry? Yes. Okay. Right. So the question is, what do triacylglycerol lipases do? And the triacylglycerol lipase is the one enzyme in the breakdown of fat that is hormonally regulated. It's called hormone-sensitive triacylglycerol lipase. And with that enzyme, the stimulation of hormone on the outside of the cell causes a phosphorylation to occur that facilitates the activation of the triacylglycerol lipase. That triacylglycerol lipase then cleaves off one fatty acid from the fat, leaving you behind with a diacylglycerol. Other lipases cleave off the other fatty acids. So the triacylglycerol lipase only works on the one that has all 3 on there. And the other lipases are not hormonally sensitive. Yes? Okay, so can I go over unsaturated fatty acid oxidation? Yes. So unsaturated fatty acid oxidation occurs as a result of a problem that arises during the oxidation of unsaturated fatty acids. I depicted two scenarios of which I only asked you to know one. And the one scenario that I asked you to know was where a cis bond during the oxidation cycle appears between carbons number 2 and 3. Between carbons number 3 and 4, sorry. I can't remember my own designations. When that happens, that substrate can't be acted on further until that double bond is moved into the right configuration. Because remember that during fatty acid oxidation, a trans double bond between carbons 2 and 3 is necessary for the further oxidation of that fatty acid. A double bond between carbons 3 and 4 prevents the formation of a double bond between 2 and 3. So fortunately, cells have an enzyme known as enoyl-CoA isomerase that converts cis 3,4 bonds to trans 2,3 bonds. And through the action of that enzyme, a trans double bond between carbons 2 and 3 is formed. At that point, you have the exact substrate that's involved in the oxidation of a saturated fatty acid, and so it can continue further, which in the next step would include the addition of water to put a hydroxyl on carbon number 3. Yes? The enzyme is called enoyl-CoA isomerase. That's E-N-O-Y-L CoA isomerase. Yes, ma'am. There is still a double bond, so it's converted from a trans 3,4 to a cis 2. I'm sorry, from a cis 3,4 to a trans 2,3. And it just continues with normal beta oxidation. That's right. Yep. That's the beauty of that enzyme. Yes, Moran. Okay, so what are ketone bodies made of? Ketone bodies are made by putting together acetyl-CoA molecules. Okay, so acetyl-CoA, as you saw in fatty acid oxidation, are molecules that are produced by fatty acid oxidation. So when the body is producing a lot of fatty acid, or breaking down a lot of fatty acids, those are energy sources because, as you've seen in class, those acetyl-CoAs can be oxidized in the citric acid cycle. Well, the problem is that the brain needs, ideally, glucose to survive, and if you don't have a good source of glucose and your blood glucose levels drop really low, then the liver will start producing ketone bodies. And the ketone bodies are a way of delivering energy to the brain with something besides glucose. So what they do is the liver does is it starts putting together those individual acetyl-CoAs. And though the actual steps aren't that critical, the important thing is that it starts with the enzyme thiolase, which is the enzyme that is the last step in fatty acid oxidation. So it runs that reaction backwards. In the backwards reaction, 2 acetyl-CoAs are converted into a 4-carbon intermediate, and then a 3rd acetyl group is added to make a 6-carbon intermediate. And then through a little rearrangement, 2 of the carbons are lost, and you're left behind with a molecule called acetoacetate. That's a molecule that has 4 carbons, and that acetoacetate is chemically unstable, meaning that it can readily decarboxylate, and if it decarboxylates, then what you produce is acetone. And it's the acetone that I said that you can smell on the breath of a person who is producing ketone bodies and can, in some cases, be an indication of poor health. Okay? Ideally, the acetoacetate is not decarboxylated, but is instead reduced and converted into a molecule called beta-hydroxybutanol, in which case the carbonyl group that's on carbon number 3 is converted to an alcohol. And that molecule is much stabler and travels in the bloodstream fairly readily. Well, however, whatever is made, what matters is getting that energy to the brain. So when the ketone bodies get made, they get dumped into the bloodstream. And in the bloodstream, they travel to the brain where they cross the blood-brain barrier. And then the reversal of all these things I've talked about happens. That is, the beta-hydroxy acetone is oxidized to acetoacetate. The CoA molecule is put back on. And those two acetyl groups are split up into acetyl-CoAs. And those acetyl-CoAs can then be used by the citric acid cycle. Make sense? Yes, sir. Okay. So the question is, in going from the 6-carbon intermediate, which is HMG-CoA, going from that 6-carbon intermediate back to the acetoacetate, is the molecule that comes off acetyl-CoA? And the answer is yes. Yep. So that's how the CoA is actually removed from the molecule, by coming off as acetyl-CoA. Well, like anything else, if I've said it in class, yes. So acetoacetate is the molecule that's unstable. Yes. Yes? Mm-hmm. Yes? It's a waste product. It doesn't. It normally just gets exhaled from the lungs, and so it's not productive for energy. Butyrate. Mm-hmm. Yeah, the acetoacetate can as well. So it's just the acetone that's of no use. Yeah. Yep. Yes, sir. Way back. Okay. Okay. No. So the question has to do with the Fisher lock and key model compared to the Koechlin-induced fit for action of an enzyme. And the question was, they both involve substrate changing an enzyme. Is that right? And the answer is no. So according to the Fisher lock and key model, all it says is that the substrate fits into an enzyme, but it says nothing about what the substrate does to the enzyme. So I think I may have described it in class as, and literally this is what the Fisher model says, is the substrate goes into the enzyme and then magic happens, and then the product is released, because it doesn't say anything about what actually happens. So the Koechlin-induced fit is a much better way of explaining how an enzyme actually works, because it does do what you said, which is that the substrate, in fact, affects the enzyme. So the example I gave in class, I think, was hexokinase, where I talked about the jaws of hexokinase, and the binding of both substrates causes the jaws to close. And so there we can see very clearly that the binding of a substrate is changing the enzyme. According to the Fisher model, all it says is that those things bind, and then that's, then you get a product. But the Koechlin model says that the binding of the substrate is what causes the enzyme to be changed, and it's the Koechlin model that also says that enzymes are flexible. Enzymes are flexible. And so it's that flexibility that allows for things like the jaws coming together to facilitate a reaction, makes that possible. Oh, yes, Callie. Sure. So chymotrypsin is a protease, and proteases are important in the process of digestion for breaking down proteins into amino acid substituents. With chymotrypsin, I talked about the mechanism by which that enzyme acted, and chymotrypsin is an example of an enzyme that is what we call a serine protease. So a serine protease is a protease specifically that uses serine as part of itself in the catalytic process. So that catalytic process involves something within all serine proteases called the catalytic triad. The catalytic triad is a series of 3 amino acids that are found in all serine proteases, and they consist of aspartic acid, histidine, and serine. And they're actually not adjacent to each other in primary sequence, but they're in fact scattered throughout the molecule, but the folding of the enzyme brings them into close proximity in the active site of the enzyme. So during the catalytic action of a serine protease, the first important step is the binding of the substrate. And the binding of the substrate causes that change in the shape of the enzyme. And it's that change in the shape of the enzyme that actually brings those 3 amino acids into closer proximity than they were before the substrate bound. Okay? So that bringing of those together causes some electronic shifts to happen. So first of all, the aspartic acid side chain is very negatively charged. And so when it gets close to the histidine ring, those electrons in the histidine ring are pushed away from the aspartic acid and in the direction of the proton of serine. And since they're negatively charged and the proton can be positively charged when it's removed, the proton comes off of serine and binds to the histidine ring. And that creates something on the serine side chain that we call the alkoxide ion. And the alkoxide ion is very reactive. It will react with the very first thing that it encounters, and since the substrate has bound in the active site, the first thing that it encounters is the carbonyl group of the peptide that it bound. So that serine makes a covalent bond to the carbonyl group of the peptide, and the binding of that alkoxide ion side chain to that carbonyl carbon causes the peptide bond to literally fall apart. And so when the peptide bond falls apart, you've accomplished and finished the first step in the catalytic process, which is the breaking of the bond and the release of the first half of the substrate. The second half of the substrate remains covalently linked to that alkoxide side chain of serine and has to be removed in order for the enzymatic process to be completed. So that happens as a result of the movement of water into the active site, and that starts what I call the slow step of the process, because it takes a while, for the relatively longer period of time for that to happen. When the water moves in, a similar thing happens with respect to removal of proton. In this case, the proton is removed from water, and the water attacks the carbonyl carbon of that peptide. That causes it to be released from the alkoxide ion, and then that peptide is released, and the entire catalytic triad is restored to its original state. So that's the sort of steps in the process. Kind of hard to do without figures. Probably harder for you guys to envision it without figures than it is for me to say it without figures. I've thought about it for many years. Yes? Okay. So anaplerotic is a term that I use to describe what the citric acid cycle is commonly described as. So the term anaplerotic means literally to fill up. And so what the term is used for with respect to the citric acid cycle is to describe how intermediates in the citric acid cycle can be used to fill up or be used by other pathways to make molecules. So, for example, there are several things in amino acid metabolism that are used either directly or indirectly from the citric acid cycle. So I described how alpha-ketoglutarate, for example, could be removed from the cycle and by transamination converted into glutamic acid or glutamate very simply. So there's an anaplerotic reaction where the citric acid cycle is supplying intermediates for amino acid metabolism. Another example I gave was oxaloacetate, which can be removed from the cycle and used to make aspartic acid, also by transamination. And the third example I gave was a molecule that wasn't actually a part of the citric acid cycle but was related to it, and that was pyruvate. And so pyruvate can be removed and converted into alanine by transamination. Now, the way that the term anaplerotic has been used over the years has been to describe it going in both ways. That is, that the citric acid cycle can fill up other pathways, and other pathways can also fill up the citric acid cycle. So when we saw the glucogenic and the ketogenic amino acids, for example, those that are ketogenic would be producing acetyl-CoA. Well, acetyl-CoA, of course, is used in the citric acid cycle, so it could be used to feed the citric acid cycle. So anyway, that term anaplerotic has been used to mean going both ways, both into and out of the cycle. In recent years, the term cataplerotic, and you're not responsible for that, but in recent years the term cataplerotic has been used to describe the movement of molecules into the cycle, and anaplerotic is used to describe the movement of molecules away from the cycle. We'll just use the term anaplerotic to describe both. Yes? Okay, so can we go over the differences between adult hemoglobin and fetal hemoglobin? Certainly. So adult hemoglobin is comprised of 4 protein subunits, 2 molecules or 2 proteins called alpha and 2 identical proteins called beta. And alpha and beta themselves look very much like each other. They're very, very closely related. And they're also closely related to myoglobin. The fetal hemoglobin, on the other hand, uses 2 proteins of the alpha, but instead of using beta, actually uses 2 related proteins called gamma. So the configuration of the fetal hemoglobin is alpha 2, gamma 2. Now the gamma is also very closely related to all of these proteins. These proteins all are called globins, by the way, which is where hemoglobin and myoglobin get their name, because the protein part of that refers to the globins. The primary differences of the fetal hemoglobin are that arise in that little donut hole of the hemoglobin. So the donut hole of the fetal hemoglobin is slightly smaller than the donut hole of the adult hemoglobin. Now that's significant because in the adult hemoglobin, the molecule 2,3-BPG, which is produced by actively metabolizing cells, perfectly fits in that donut hole of the adult hemoglobin. Well, the effect of 2,3-BPG on the adult hemoglobin is it converts it from the R state, which is a tight binding state, into the T state, which is a loose binding state, which causes the hemoglobin to tend to release oxygen. In the case of the fetus, fetal hemoglobin does not bind 2,3-BPG. So it doesn't readily flip from the R state to the T state nearly as readily as the adult hemoglobin does, and consequently has a greater affinity for oxygen than the adult hemoglobin does, which is how the fetal hemoglobin actually takes the oxygen away from the mother's adult hemoglobin and then uses it within the fetus's body. Does fetal hemoglobin exhibit cooperativity? Yes, but it's a little bit different, as you can imagine. So when we think about cooperativity, cooperativity happens in the lungs where the oxygen concentration is really, really high. And there's not a great concentration of oxygen, of the oxygen coming in from mom. So if the question is, can it exhibit cooperativity, the answer is yes. But it's not as pronounced of an effect as you see in the adult because there's just not as much oxygen. Yes. Okay, so the question has to do with the 2,3-BPG and a smoker. and whether or not the 2,3-BPG has the effect on the fetus's ability to carry oxygen. Is that safe to say, Andrew? Okay. Well, the answer is that 2,3-BPG has no effect on the fetus's ability to carry oxygen. And that's because 2,3-BPG does not fit into the fetal hemoglobin. There is an effect, and the effect is that when mom is smoking, her oxygen carrying capacity is lower. So because her oxygen carrying capacity is lower, the fetus is going to be more starved for oxygen than mom is. If it gets the oxygen, it's fine, but it has to get enough oxygen from mom to be able to, go through the respiration that it does. So the only effect on the fetus is due to the mom's reduced oxygen carrying capacity, just like any smoker. Okay, but the fetus itself doesn't have any effects of the 2,3-BPG from the smoking. I've been cleaning my garage today, so I hope I'm not too grubby standing up here. Yes, Anna? Yeah, it's a good question. I didn't say where they came from. So her question basically is, where do HDLs come from? HDLs are actually manufactured by the liver. Just like the liver manufactures the VLDLs, which break down and become IDLs and then LDLs, the liver also manufactures the HDLs. You're not responsible for that, and that's actually kind of a complicated process. But they don't come from the chylomicrons. No, they don't. Yes? Can I go through the glyoxylate cycle? Sure. So the glyoxylate cycle, as you may recall, is a cycle that is sort of overlapped or overlaying the citric acid cycle. So let me at least draw this one on the board for you. So let's do that and show you a little bit. Oops. Freedom to ask nothing, expect nothing, to depend on nothing. Okay, well. All right, so if we look at the citric acid cycle, we have, of course, the starting point of the acetyl-CoA. Oops. We have the oxaloacetate. We have the citrate. Citrate goes to isocitrate, goes to alpha-ketoglutarate, goes to succinyl-CoA, goes to succinate, goes to fumarate, goes to malate, goes to oxaloacetate. So that's a citric acid cycle right there, okay? There are two places in the cycle that involve decarboxylations. One place in the cycle is right here. Carbon dioxide is released. Another place in the cycle is right here. Okay? Now, the citric acid cycle is related in that it uses several of the same enzymes. But when the cycle gets to isocitrate point, there's an enzyme that the plants and yeast and bacteria have called isocitrate lyase. Okay? And so isocitrate lyase produces succinate and it produces acetyl-CoA. Produces glyoxylate. Okay. Addition of another acetyl-CoA to the glyoxylate yields malate. And this is catalyzed by the enzyme malate synthase. Okay. So these two enzymes are necessary to run the glyoxylate cycle. They're only found in plants, bacteria, and yeast. And when they do this, the decarboxylation reactions of the citric acid cycle are lost, meaning that there's no decarboxylation. So you start here, you go around through here, and remember that this succinate that was made down here moves up the pathway so that by the time you get here, you've got two oxaloacetates. And that's different from the citric acid cycle, where we started with one and we ended up with one, which means that at the end of the glyoxylate cycle, or the completion of the cycle, I shouldn't say the end, but at the completion of the cycle of the glyoxylate cycle, an extra oxaloacetate is produced. And that oxaloacetate can go to gluconeogenesis. It can go to amino acid synthesis. It can go to a variety of things, okay? And the point is that the cycle is kicking that out each time it goes around. Now, when does the plant or the bacterium use the glyoxylate cycle, and when does it use the citric acid cycle? It's really a function of how much NAD it has. So when the cell is low on NAD, the reactions that require NAD are this one, and this one. So the glyoxylate cycle actually needs NAD. It needs 1. It doesn't need 3. So the glyoxylate cycle doesn't have as high of a demand for NAD as the citric acid cycle does. And so when NAD concentrations are low, the glyoxylate cycle will be favored. And when the NAD concentrations are high, then the citric acid cycle will be favored. Okay? Yes, sir. Mm-hmm. So the question has, if I understand it correctly, the question is, will a need to make amino acids drive the glyoxylate cycle to run? Is that what you're saying?
Yeah, and vice versa, like is regulated by
The NAD is the primary regulator, yes. So it's an energy consideration. If you think about it, when NAD concentrations are low, what does that mean? It means that NADH is high. And NADH is high usually happens when cells are fermenting, because they have the ability to go back and convert NADH back to NAD. So fermentation would be a time when you would expect that you might see a greater increase in the glyoxylate cycle. And one of the ways that you could deal with the byproducts of the glyoxylate cycle would be to do either of these: make more glucose, or, as you note, make, in this case, aspartic acid. Yes, sir. Purines, okay. You're talking about the metabolism of purines, is that what you're asking about? Okay. So the question had to do with the metabolism of purines. So when we talk about the metabolism of purines, we're talking about how it is that adenine, guanine are either made or broken down. And I shouldn't say adenine or guanine, but adenosine or guanosine, how those are made or broken down. Both of these molecules, as I noted in class, are made from very simple starting compounds. So there's some amino acids, some carbon dioxide, and some other things that go together to make the ring structures of the purines, which is that 2-ring, 6-molecule, 5-molecule ring structure that I showed you. I didn't talk about the early stages of that synthesis, but I did point out that there's an intermediate that is a branch point between the synthesis of AMP and GMP. And that intermediate is known as IMP. And IMP is known as inosine monophosphate. You actually saw that in class one time during the term. Does anybody remember where they saw it? The wobble position. Yeah. So inosine actually appears in the wobble position of a tRNA molecule. All right. In any event, the important thing for our purposes is that IMP is a branch point. So one branch leads to the synthesis of AMP, one branch leads to the synthesis of GMP. It turns out that AMP feedback inhibits its own synthesis. So that first reaction that goes from IMP is inhibited, that enzyme is inhibited by AMP. I noted that the synthesis of AMP requires energy from GTP. And that makes sense because if cells are making AMP, it means they've got too much GTP and not enough AMP. It could mean that. I shouldn't say it does mean that, but it could mean that. The synthesis of GMP is similarly feedback inhibited by the appearance of GMP. So GMP will inhibit the first enzyme catalyzing the reaction from IMP in the direction of GMP. And as I noted in class also, ATP is the energy source for making GMP for the same reasons that GTP was the energy source for making AMP. So once you've made the GMP and the AMP, you convert them into diphosphates using the nucleoside monokinases or monophosphate kinases, which converts the GMP and the AMP into ADP and GDP. And then the enzyme nucleotide diphosphokinase, or NDPK, is the enzyme that converts the diphosphates into the triphosphates. And I noted that that enzyme converts all of the diphosphates into all of the triphosphates. On the breakdown side of the purines, where a cell has too many purines, they go through an intermediate known as uric acid. And uric acid is the molecule that I described that causes gout. And it causes it because uric acid is relatively insoluble in water and in high concentrations, which would arise during the breakdown of a lot of purines. It will crystallize out of solution, and it'll precipitate usually in the lowest part in the body, which is in the big toe. And so it hits a nerve cell there and causes big problems. So that's really what I said about those. The only other thing I mentioned relative to the metabolism of purines had to do with the salvage of purines. And I said that there was an enzyme called HGPRT that was deficient in people who had a syndrome known as Lesch-Nyhan syndrome. That's LESCH-NYHAN. And that was the syndrome where people chewed their own lips and fingers off, which was, yeah. Okay? Yes? Okay, so good question here. Why, since the balance of nucleotides is so important, why is ATP so abundant everywhere? And the answer to that question is, it's a very good question, actually. ATP is the triphosphate that's in the highest concentration in cells. And it turns out that the enzymes, the RNA polymerase and the DNA polymerase, has evolved to be able to handle that higher concentration. Which it pretty much has to be able to do. Otherwise, you would see some effects arising from that. Now, the other nucleotides are, in fact, used for other energy purposes. They're just not as widely used as ATP is. So I mentioned that GTP was used to drive protein synthesis, that CTP was used to drive the glycerophospholipid biosynthesis, and UTP was used to synthesize glycogen. So all three of the other nucleotides are, in fact, used to make things. They're just not quite as widely used as ATP is. And by the way, when I say that balance is important, it doesn't mean that they're all equal, but it means that there's a range where that they're acceptable in order to be in it. So that's why that evolution of the enzymes to be able to handle that is important. Yes, sir? How do you fix gout? Yeah, you treat it by giving people a molecule called allopurinol. And allopurinol inhibits the production of uric acid. And so what allopurinol does is it favors the salvage back that is remaking the purine nucleotides. So it's an inhibitor of the enzyme that produces uric acid is what it is.
So when you have that dissociation,
Once it's formed, you're going to have gout. Yeah, that's going to be there. But it's not there forever. So it's important to realize that it crystallizes because it's precipitating out a solution. If you change the concentration of uric acid slightly by reducing it, let's say with allopurinol, then it's just going to redissolve and you're going to be fine. Yeah. Yes?
Do you repeat the enzymes that convert the
Yeah, the enzymes that convert the monophosphates into diphosphate, they have a variety of names. So I can't remember what I had on the figure that I showed in class, but I call them nucleoside monophosphate kinases. Okay, they might be called nucleotide monokinases, something like that. Okay, those names are the reason that the name isn't really set is each one has its own name, and there's one specific for each of the individual monophosphate kinases. But the one that does all the diphosphates is nucleoside diphosphokinases, NDPK. That one does have a fixed name. Yeah. I shouldn't say that. It actually has. It's called nucleoside diphosphate kinase. It's called nucleoside diphosphokinases. It's called nucleotide diphosphate kinase. So there's quite a few names for that one as well. But that's only one enzyme. Yes, Andrew? There is one for each of the monophosphates, yes. The diphosphates are just one, yeah. Yeah. My garage is looking a lot better. It's needed cleaning for a long time. You know, there's something very satisfying about throwing things away, taking things to Goodwill, and then having a nice, organized facility after you're done. I've been working on a couple of sheds for the past couple of weeks, and so now I have 2 clean sheds and 1 clean garage. So I'm a very happy guy, Nick. So. Happy enough to write an easy exam. I am, yeah. Well, I've already done it, but thank you. Yeah. So what are the averages usually like for the final? Given with this class, it's been very hard to predict what's going to happen. So I'm a little reluctant to tell you what I'm going to tell you, Mariah, but I'll tell you anyway since you've asked. The average on the final usually falls. And it usually falls... Hear me out here. It usually falls because, the average on the earlier exams are usually larger than what you guys have had here. And I think that it's because everybody's got other exams during that week. So it's just a lot of stuff that's there, okay? One of the things that I've seen with low averages over the years, and this is pretty constant, is that when a class has a very low average, which is what you guys had on the last one, the next one they almost always bounce back because I think people get worried and perhaps spend more time or make sure that they get their questions answered and so forth, perhaps more than they had done before. So I've been very pleased with the interactions that I've had, and every indication I see is that it looks like people are going to do better. And so I'm very hopeful for that. And the other thing that you have on the final exam is you have that note card. And that note card helps you to organize things, both your thoughts and also to do things like saying, wow, I don't remember the name of the enzyme that Ahern talked about with the glyoxylate cycle. So I'll write it down on my card, okay? And then I put it out of my head. I don't have to think about it again. I just have to know where I put it on my card so I can remember something like that. So I'm optimistic about the final exam. And remember that it's a curved exam. So, well, you say it's the scary part. I don't think so. I think, what's that? So let me just tell you a little bit about grading and sort of philosophy of grading, okay? So if you look at biochemistry, you say, oh, it's a really, really hard subject. And so if the really hardness of the subject were a critical factor, you would see a greater percentage of F's and D's than you would see in other classes. But you don't, okay? So where the average goes down, the grade goes down as well, right? That is that what it takes to make a C goes down, all right, if the average goes down. So what matters is not how hard the exam is, or for that matter, even what the average in the class is. What matters is where you do relevant to that average. That's what matters. And if you track that, and I do track that, you'll see that most people don't change things drastically from the average. And so if you see where you did exam 1 and exam 2 relevant to the average, you probably saw that exam 3 went down, but if you compared it to the average, it probably didn't change that nearly as much compared to the average. So yes, Mariah?
So mine didn't like after the main grade, and it's like even if it's better than the average, I didn't have as much chance to make that than even
Yeah. So it's not exact. I mean, I won't say that if you made 4 points below the average on the first one, you're going to make 4 points below it on the second one and the third one. That's not what I'm saying. But in general, they tend to follow where the average goes, right? Because that's what the average is, the whole class, the class as a whole, right? So yes, some people will go up, some people will go down. There were probably about 4, maybe 5 people I talked to who had significant jumps on the third exam. So it wasn't that everybody went down. So if you had a significant jump on the third exam, what do you suppose happened to your overall grade compared to the class? It went up, right? And so the best thing that you can do is improve your standing relevant to the average. And that's why I always tell students, the thing that's in your best interest is not for me to make an easy exam. It's for me to make a murderous exam. So that the lower the average is and the better you do against it, that's how you improve your grade. I'm not going to go make a murderous exam because people get all kinds of anxious about that. But the reality is that that's the way that students actually improve their grades against that. Make sense? Yes?
I know you mentioned this in class, I don't think I wrote it down. The percentage of material from the different
Yeah. So what's the percentage of material from the different units? So roughly, each unit is represented on the final by the same percentage as it was during the term. And so that means for the new material on the exam, it's roughly 25 to 30%. And then each of the other parts are roughly 25% as well. So because we've had exams on average about every 2 to 2.5 weeks during the term, there hasn't been one that's had a lot more number of lectures compared to a different one.
Would you say the way you write the test, you would avoid similar material that we've been tested on in past tests?
So would I say that I would avoid similar material that you've been tested on in past tests? No. I would say that I would avoid using the same questions. And there's even an exception to that. There's usually 2 or 3 questions that you'll see that are identical to the previous exam because I felt like, well, this was an important thing that I think people should know. But you'll see the same topics. That's why I recommend using the older exams as an outline for your studying, because those topics I said were important, and those topics are important for the final exam. Yeah. Yes, sir. The fish membrane. Oh, yes, I do. Yeah. Sure. So the principle here, the question had to do with the fish membranes and the sort of biochemistry of those that allowed them to stay fluid in the cold environment of the ocean. And the answer is it's a very basic, simple principle, and that is that if you recall when I talked about this melting temperature or this conversion temperature from a more solid-like to a more fluid-like character, that was largely a function of how unsaturated the fatty acids in the lipid membranes were and how short they were. So the more unsaturated and the shorter they are, the more fluid they will be at colder temperatures. That is, the lower their melting temperature will be. You don't want your membrane solidifying, basically, is what I said. The other part of that question asks people, what are the two groups of molecules that are most commonly found in lipid membranes? And that was unfortunately missed by a lot of people. But I talked about those two groups of molecules as glycerophospholipids and as sphingolipids. Cholesterol is not a group of molecules, correct? Cholesterol is a single molecule. Proteins, not really an answer, because proteins are found in a wide variety of places besides membranes, and so that we didn't take that answer either. Andrew? Oh yes, cholesterol is definitely associated with the membranes of every organism, or certainly every animal. No. So the question is, do cholesterol make membranes more fluid? And the answer is not really, okay? At least not as reflects temperature. So what cholesterol does is it widens the transition temperature over which that transition from a more solid-like state to a more fluid-like state occurs. So if you want to call that widening of that range making it more fluid, then I guess that's okay. But it doesn't have any effect on the temperature. That is, the midpoint of that stays exactly the same with cholesterol. Now, one thing I didn't get to tell you guys, I'm going to take a minute here and tell you, because this is something I find students always like. You're not going to need to know this. But I promised I would talk to you about a photosynthetic fish, and I didn't get to do it. So I want to take a minute and tell you how to make a photosynthetic fish, okay? And you guys know enough to make a photosynthetic fish. So my idea goes as follows. You know those glass fish that you can buy in an aquarium store, right? You can see through them, essentially. So they're transparent. So my idea involves this transparent fish that you can get in an aquarium store. And it involves a protein that I talked about in class called Bacterial Rhodopsin. So Bacterial Rhodopsin, you might remember, was a molecule, was a protein that I said that contained a molecule of vitamin A in its center. And the molecule of vitamin A in its center does the following thing. It kicks. You remember the kicking that I talked about in class? Somebody says yes, somebody says no. Okay, well, I did. So it turns out that vitamin A in the center of this protein, there's a chamber. In the very middle of that chamber, there's a molecule of vitamin A. And vitamin A is light sensitive. Okay? So what happens is when you shine light on this protein, the vitamin A goes from all trans to cis to all trans to cis. And it goes back and forth very much like my foot does. Well, the effect that has in the membrane of a bacterial cell is every time it kicks a proton out of the bacterial cell and creates a proton gradient on the outside. That's one of the ways that photosynthesis can occur in bacteria. Okay? By creating a proton gradient, you know that a proton gradient can be used to make ATP when the protons come back in, right? Everybody understands that, right? So I say we take this protein and we put it in the mitochondria of these glass fish. Now, what happens when we shine a light on these glass fish? Well, start isomerizing, start kicking protons out, create a proton gradient, and what does a mitochondrion do with the proton gradient? It makes ATP, right? This is what happens in photosynthesis in plants. Photophosphorylation, light creates ATP, right? So this could make a proton gradient needed to make ATP. Now, it's not truly a photosynthetic fish in that the dark cycle of photosynthesis would not occur because there's not a way for your mitochondria to assimilate carbon like happens in the Calvin cycle. However, you would have a very, very new source of energy in the form of light, okay, that could be used to make ATP, and so consequently the amount of food you would have to give to such a fish would be very, very small. Make sense? It would be more like a solar-powered fish. I like that. I like that, actually. No. So the ATP would be on the inside just like before. It's the proton gradient would be exactly the same. It would be exactly the same driving force. All that you would have is instead of electron transport kicking the protons out, you would have light kicking the electrons out. Or the protons out, rather. Yeah. So would you have to embed it in a certain part of the mitochondrion? Well, it's always hard. I mean, since we're speculating, we can speculate. And I would say I don't think it would require that. I've actually talked to somebody who's a mitochondrial expert about this idea, and he said he thought it sounded feasible. So I tell this to my students. I've been telling this to students for years, saying, if you ever work in a lab, talk to your professor and see if they'll let you make a photosynthetic fish. And I've actually had several students, in fact, who have spoken to the professors, and at least one I heard from last year was trying to do this experiment. It'd be kind of cool to do. So if you do it and you make $1 billion, you know where you heard it first, right? Just remember me in your will. Yes, sir. The two what? Glycerophospholipids. The question was, what are the two classes of molecules in lipid membranes? So glycerophospholipids and sphingolipids. Yes, Maura. Names of the molecules, yes. Sure. So there's only two, and they have the same coenzymes. So one was the pyruvate dehydrogenase, which isn't part of the cycle, and the other is the alpha-ketoglutarate dehydrogenase, which has the same coenzymes. And there's 5 of them, so yes. All right, guys. Well, good luck. I will be in my office, so come see me if you have questions. Good luck on the exam, and let's see you improve against the average, right? That's the aim.